Hello, I'm Dr. Helen Warren and I'm a lecturer in statistical genetics at Queen Mary, University of London in the UK. And we're describing to you our recent publication in Nature Genetics titled Genome-wide Association Analysis Identifies Novel Blood Pressure Loci and Offers Biological Insights into Cardiovascular Risk. We used a discovery sample of just over 140,000 individuals of European ancestry from the UK Biobank cohort and tested almost 10 million genetic variants across the genome for association with blood pressure, followed by replication in large independent datasets. We identified and robustly validated 107 independent genetic signals associated with blood pressure, which were not previously reported. And together, these highlight new biological pathways for blood pressure regulation and suggest potential new drug targets for the treatment of hypertension. The primary genetic association analysis used linear regression within SNP test software. We also carried out a range of computer-based bioinformatics analyses and laboratory experiments. For example, pathway analysis to identify the most important biological pathways involved in blood pressure regulation, and also gene expression testing, which showed that blood pressure genes are most strongly expressed within vascular tissues. Combining all the genetic variants associated with blood pressure together into a genetic risk score, we show that altogether they increase systolic blood pressure by up to 10 millimeters of mercury in adults over 50 years old. This is comparable to a blood pressure reduction that could be achieved by improving lifestyle factors alone. Hence, we recommend further study into a possible precision medicine approach for the future, which could adopt earlier lifestyle intervention amongst individuals of high genetic risk to offset the impact of their blood pressure raising genetic variants on their future cardiovascular risk. As a statistician, I think our analysis shows the great benefits of using data from cohorts like the UK Biobank for enabling discovery of many new genetic signals associated with diseases. There are three main advantages. 1. Having large sample sizes within one single cohort. 2. Having standardized phenotyping where all participants have had blood pressure measured in exactly the same way. And 3. Having high quality genetic data using modern genetic imputation methods to cover millions of genetic variants across the genome.